Hello everyone, I'm David Farzam. Welcome to New Cancer Mentality. This is part 3 of the 3-part series, Cancer in the Post-Genomic Era. Where do we go from here? What will it take to prevent, cure, or control cancer? In this section, Dr. Glazier will discuss requirements for the cure or control of cancer and propose practical ways to bypass the staggering genetic complexity of the disease and specifically cure or control metastatic cancer. He will also talk about a proposed approach for the treatment and cure of brain cancer. It's great to have Dr. Arnold Glazier back on the New Cancer Mentality website.
Thank you. It's nice to be back.
So Dr. Glazier, why is it important to understand the requirements for the cure or control of cancer?
The requirements sharply define all possible solutions to the consistent cure or control of cancer. By understanding the requirements, we can know what will not work and what cannot work. If a process or therapy cannot satisfy the requirements, it will fail. It may prolong survival by weeks or months, but it cannot consistently cure or control the disease. An understanding of the requirements places cancer research on firm ground. It allows us at long last to develop a scientifically sound strategy for the consistent cure or control of metastatic cancer. Within the context of the requirements to be discussed today, the cure and control of cancer without significant toxicity to the patient becomes a solvable engineering task.
Dr. Glazier, how can there be only one set of requirements that must be satisfied to cure or control all types of cancer? As you pointed out in part 1, cancer evolves. Every patient's cancer is different and every cancer cell is different, even in the same patient.
There is a common set of requirements because the problem that needs to be solved is the same for all patients with cancer. The comprehensive detection and destruction or control of an enormously diverse, unpredictable, evolving population of cancer cells in the patient.
Can you briefly review the 3 basic conditions that you talked about in part 1 that need to be satisfied to cure or control cancer?
Sure. Any process that can cure or control cancer consistently must jointly satisfy 3 basic conditions: comprehensiveness, specificity, and knowability. Comprehensiveness refers to the need to kill or control all malignant cells in the patient. Specificity refers to the need to kill or control cancer cells without excessive toxicity to the patient. Specificity depends upon differences between cancer cells and normal cells. And knowability refers to the need to target properties that can be known or accurately predicted. Unknown or unpredictable properties cannot be targeted.
Are these logical requirements for the cure or control of cancer?
There are logical requirements for the cure or control of cancer. There are certain logical steps that any process or mechanism for the specific cure or control of cancer must carry out. These required steps involve asking questions about the cells in their microenvironment, receiving bits of information in the form of answers, deciding normal cell or cancer cell, and killing only the cancer cells. If you were writing a computer program to detect and destroy cancer cells, these would be the key steps required for the program to work. And this is true not just as a matter of logic, but also as a matter of physics. A process that could specifically cure cancer without engaging in these steps, which involve information, would violate the second law of thermodynamics and be akin to creating Maxwell's demon. A logically equivalent or isomorphic process must be carried out by any drugs or anti-cancer therapies that can specifically cure or control cancer. These requirements are about information, principally the information required to detect and destroy all malignant cells that could evolve. The minimal set of questions that must be asked and answered to accomplish this task define the information requirements for the cure of cancer. As we will discuss later, the stochastic evolutionary nature of cancer sharply defines the questions that must be asked and answered by any process that can specifically cure or control cancer. I know this sounds very abstract, but when these requirements are translated into the language of molecular biology and chemistry, they become very concrete.
What does it mean to translate these requirements into the language of chemistry and molecular biology?
It means developing isomorphic chemical and molecular processes that involve the same information and meaning, that are structurally equivalent, and in a sense map into these requirements. It means designing and developing drugs and anti-cancer technologies that can carry out these processes. Tens of millions of chemicals have been screened for anti-cancer activity with almost no success. The chance of discovering drugs and therapies that meet these requirements is essentially zero. The drugs need to be purposefully designed. But before we can do that, we need to redefine the problem of cancer and put it into a form that is solvable. As currently formulated, the problem of cancer is unsolvable.
Where would you like to start?
Let's begin with a very basic question that defines the problem to be solved. What population of cells must anti-cancer therapy be able to target, be able to kill or control for the consistent cure or control of cancer? The obvious answer is all cancer cells that are present in the patient. However, this answer is false. It presupposes knowledge that generally cannot be known. If we could know all that has evolved and know all that will evolve in a patient with metastatic cancer, then at least in principle we could target it, but we cannot know. So in order to achieve comprehensiveness, we must target all cancer cells that could evolve. This is a really important point. The set of all malignant cells that could evolve is the cell population that must be targeted for the consistent cure or control of cancer. Therapy that targets a lesser set or subset will fail.
In an earlier webcast, you stated that at least 10 to the 68,000th power of different types of cancer cells could evolve.
Yes. This means we must shift our focus from targeting properties of cancer cells to targeting properties of the set of all malignant cells that could evolve. The two are very different. Pathways of tumor cell evolution are properties of cancer cells, not properties of the set of all malignant cells that could evolve. For decades, it has been assumed that therapy for the cure or control of cancer should be dependent upon the particular pathways of tumor cell evolution. This was the rationale for the massive cancer genome sequencing project. This assumption is no longer tenable. It is inconsistent with a vast amount of experimental evidence. It is inconsistent with the fact that cancer is an incredibly diverse stochastic evolutionary process. It is false. Targeting pathways of tumor cell evolution can provide specificity, but generally not comprehensiveness. The essential features of any process that can consistently cure or control cancer must be independent of the pathways of tumor cell evolution. To summarize, the required target for the cure or control of cancer is the set of all malignant cells that could evolve. The properties of individual cancer cells are not relevant to the cure or control of cancer. The properties of subsets of cancer cells are not relevant. What matters are the properties of the entire set of all malignant cells that could evolve. This redefines the problem in a way that reflects the reality of nature, that cancer is a stochastic evolutionary process. As I mentioned in part 1, and we'll discuss in detail, we need a higher level theory of cancer, based not on the properties of cancer cells, but based on properties of the set of all cancer cells that could evolve.
The concept of the set of all malignant cells that could evolve seems a bit vague and ill-defined. Is there a black and white line between what could evolve and what could not evolve in a patient with cancer?
No. We're dealing with probabilities. Consider the universe or set of all possible malignant cells, with all possible combinations of genetic and epigenetic alterations. Given unlimited time, any of these cells could evolve from a normal cell in the patient. However, time is short. Malignant cells that are actually present in the patient, by definition, have a probability of 1. By contrast, there is some type of malignant cells that is so improbable that for all practical purposes their probability is 0. In between these extremes, there would be some type of probability gradients.
Can you know the probability that a particular type of malignant cell will evolve?
No. Any mathematical function that describes the probabilities of what could evolve must generate output that has stochastic or random features. This follows from the many different layers of stochastic complexity of tumor cell evolution. The bottom line is, probabilities that describe what will evolve in a patient with cancer are unknowable. We need to look at the extreme cases, the limits. What is so improbable that it will never happen? What is so probable that it will always happen? These are the limits or constraints. It is at the extreme limits that we can have almost absolute certainty. This is how we must approach the problem of tumor cell evolution. The target set must be very broad and extend to cover the extreme limits of improbability, where there is near certainty that no malignant cell could evolve that is not a member of the targeted set.
Doesn't this just replace the problem of targeting what has evolved with the even more difficult problem of targeting what could potentially evolve?
No, it does the exact opposite. It vastly simplifies the problem. The nearly unlimited genetic and epigenetic complexity of cancer no longer matters. The unpredictable pathways of tumor cell evolution no longer matter. The fact that every patient's cancer cell is unique, that every patient's cancer is unique, no longer matters. The fact that every cancer cell is different no longer matters. The fact that cancer cells can't evolve to resist to drugs no longer matters. They all become irrelevant to the cure and control of cancer. What matters are the properties of the set of all malignant cells that could evolve. These properties are independent of the pathways of tumor cell evolution. Furthermore, the set of all malignant cells that could evolve is essentially the same for all solid cancers and all patients. Therefore, at an important level, cancer is essentially one disease. Anti-cancer drugs that target the set of all malignant cells that could evolve would be able to cure or control all types of solid metastatic cancers: breast cancer, colon cancer, pancreatic cancer, ovarian cancer, kidney cancer, melanoma, to name just a few. Furthermore, to consistently cure metastatic breast cancer, a therapy would also need to be able to cure cancers such as colon cancer, prostate cancer, and pancreatic cancer. It's really all the same problem: the detection and destruction of the set of all malignant cells that could evolve.
Prostate cancer is usually slow growing and harmless, while pancreatic cancer is usually rapidly growing and fatal. So how can cancer be one disease? How could the same drugs you propose cure or control all types of cancer?
Every patient's cancer is different. What actually evolves is different in every patient. What actually evolves in a patient with prostate cancer is different from what actually evolves in a patient with pancreatic cancer. But, and this is the important point, what actually evolves is a subset of the set of all malignant cells that could evolve. Targeting what could evolve would comprehensively target what actually evolves in all patients with all types of cancer.
The set of all malignant cells that could evolve is an abstraction. It does not exist in the real world. It cannot be observed. It cannot be experimentally characterized. So isn't this outside the scope of science?
Absolutely not. And this is a very important point. It's pointed out by the philosopher Karl Popper. Observation and experiment are not the logical source of scientific knowledge. Scientific knowledge comes from hypothesis, deductive logic, and refutation. The role of empirical data is as a means of refutation, to disprove hypotheses. A subject matter is within the scope of science if it is possible to formulate a theory about it that could be falsified or proven wrong by empirical evidence. We can formulate a proper scientific theory about properties of the set of all malignant cells that could evolve. Observation of a single malignant cell that contradicted the theory would disprove it. As a matter of logic, no amount of experimental data or observations could ever prove any theory to be true, even if it is true. The truth of a scientific theory is always provisional. As we continue, we shall see that it is possible to formulate a scientific theory about the set of all malignant cells that could evolve that resists the severest attempts to prove it false. A theory that is true, but like all scientific theories, cannot be proven to be true. By contrast, as a matter of logic, it is not possible to formulate a true theory of cancer based on properties unique to a subset of all malignant cells that could evolve, because cancer cells can evolve without those properties that would prove the theory false.
What can be known about the set of malignant cells, all malignant cells that could evolve?
Very little, but enough. What can be known sharply defines the requirements for the specific cure and control of cancer. Let's begin with some definitions. The set of all malignant cells that could evolve have two defining properties. All the cells are malignant, all the cells could evolve. Malignant behavior, proliferation, and invasiveness in an abnormal context is the defining property of malignant cells. The component functions of malignant behavior, proliferation and invasiveness, need not occur at the same time and usually don't. However, the potential for cell proliferation is a constant, time-independent property of malignant cells. Most malignant cells do not engage in malignant behavior most of the time, but at some point in time all must, otherwise they're not malignant.
Is cell migration the same as invasiveness?
No. Invasiveness is the expansion of cells into new space with the destruction of existing tissue architecture and the creation of new infrastructure to support the metabolic needs of the cells. It's important to recognize the concept of cancer cell is actually a bit murky and ill-defined.
What do you mean? Why is the concept of a cancer cell murky and ill-defined?
Because the defining property of cancer cells, malignant behavior, is not a property solely of the cell. It's a property of the cell and its microenvironment. It's impossible to examine a cell in isolation and determine if it is a malignant cell. The same cell can behave totally different in different sites of the body. In one environment, the cells can remain dormant, and in another site in the body, the cells can cause large invasive cancers. Furthermore, while proliferation is a property solely of the cell, invasiveness is not. Invasiveness is a property of the cell and its microenvironment. It can be a cooperative process involving many cancer cells and normal cells.
What else can be known about the set of all malignant cells that could evolve?
I'll state it in the form of a scientific theory. All malignant cells use normal cellular machinery to engage in malignant behavior. And this normal machinery that carries out malignant behavior can be expressed by malignant cells and also by non-cancer cells in the microenvironment that aid in the process. An exception to this theory has never been reported, and we can be confident that an exception will never arise. There is not sufficient time for the evolution of extensive new functional machinery. The complexity of the requisite interdependent biochemical processes is too great. The joint probabilities are too small, and the human lifespan is far too short for a tumor cell to evolve that violates this theory. This brings us to a very important point. The nearly unlimited genetic and epigenetic chaos of cancer is constrained. What could evolve is constrained. A malignant cell will result if and only if the genetic and epigenetic alterations cause normal cellular machinery to carry out the processes of proliferation and invasiveness. Genetic and epigenetic lesions that do not fulfill this criteria result in dead-end tumor cells that are not malignant. This imparts a profound simplicity to cancer. The nearly unlimited genetic and epigenetic complexity of the disease becomes irrelevant. What matters is the normal cellular machinery that could carry out malignant behavior.
But new cellular machinery does evolve in cancer cells.
Yes, new functional cellular machinery certainly does evolve. The BCR-ABL protein is a good example. The new machinery that evolves, which contributes to cancer, does so by causing normal cellular machinery to carry out malignant behavior. Furthermore, only a limited amount of new functional cellular machinery can evolve. There just isn't enough time for extensive amounts of new functional machinery to evolve. Violation of this rule is about as likely as a healthy person being born without a brain and a heart and having in their place totally different organs.
Earlier you described the nearly unlimited genetic and epigenetic complexity of cancer. Now you were saying that there are limits to tumor cell evolution and limits to what could evolve. Isn't this somewhat contradictory?
No. Tumor cell evolution is incredibly complex, but is severely constrained by time and history. Evolution is actually very much more about conservation and stability than about change. The evolutionary journey from normal cell to cancer cell typically involves less than several thousand cell divisions. That's enough to generate astronomical tumor cell diversity and to enable resistance to virtually any single cancer therapy. But it is not enough time to create extensive new cellular machinery. It is not enough time to revise or replace large numbers of essential, evolutionarily conserved genes and proteins that have survived for hundreds of millions of years or more. Cancer is evolutionary but constrained. Tumor cell evolution has limits. The fundamental cause of these limits is the short time available for tumor cells to evolve. But there are other factors that make evolution a conservative, gradual process.
Can you explain in more detail why evolution is a conservative, gradual process?
Evolutionary systems in nature are extraordinarily complex and involve millions of interacting parts that function together to promote self-survival and reproduction. There's an almost unlimited number of ways that millions of components can be randomly assembled. Most configurations will be non-functional. The evolution of complex functional machinery is necessarily a very slow, gradual process of trial and error, mostly error. It takes a long time and a lot of generations to get it right. In addition, evolution is a cumulative process. Evolution is about modification of existing life. Extinctions occur, but all known life on Earth is related. This gives an evolutionary layering of complexity. New components must fit and interact in a functional manner with existing machinery and provide a selective advantage. The interdependency and interconnectedness of biological systems makes it difficult to change some existing components. It's like the pieces of an interlocking puzzle. To change the shape of one piece requires altering the shape of neighboring pieces. This in turn could require changing the shape of their neighbors and set off a kind of chain reaction, all of which is improbable when the source of change is random variation. Some cellular components are so important and interact with so many other critical parts that their change is almost impossible without impairing survival. However, redundancies and backups can and do exist, so it should not surprise us when seemingly essential proteins are lost or mutated. We'll return to the topic of redundancy later when we talk about the requirements for the cure of cancer. For now, the important point is that tumor cell evolution is constrained by the requirement that the underlying genetic and epigenetic alterations cause normal cellular machinery to carry out proliferation and invasiveness. What matters is the normal cellular machinery that could carry out malignant behavior.
What do we currently know about the normal cellular machinery that could carry out malignant behavior?
The set of normal cellular machinery that is required for or characteristic of proliferation and invasiveness is fixed, finite, and encoded in the normal human genome. The machinery is represented in the biochemistry of DNA synthesis, cell division, wound healing, blood vessel formation, placental growth, and fetal development. The set of normal cellular machinery that could carry out malignant behavior is the same for all types of cancers, independent of the pathways of tumor cell evolution, comprehensive, knowable, and is largely already known.
Are you saying that all malignant cells use the same normal cellular machinery to engage in their malignant behavior?
No. Differences between cells are well known. For example, the protein urokinase is involved in the process of invasiveness. Some malignant cells will express urokinase and some will not, even in the same patient. Under the proper selective conditions, urokinase positive cancers can become urokinase negative and vice versa. The normal cellular machinery that actually carries out malignant behavior in a particular patient, in a particular cancer cells, is dependent upon the pathways of tumor cell evolution. It is unpredictable and unknowable unless we examine every single cancer cell in the patient, which is at least a practical impossibility. What I'm saying is this: there exists a finite set of normal cellular machinery that carries out proliferation and invasiveness in normal physiological processes, such as placental implantation and wound healing. All malignant cells that could evolve will use elements of this set of normal cellular machinery to engage in malignant behavior and can be detected on this basis.
There are thousands of different normal proteins involved in the process of cell proliferation and invasiveness. How could we target or inhibit all of this? Aren't we once again drowning in the complexity we were in beforehand?
No. To cure or control cancer, we do not need to inhibit or target all the normal cellular machinery involved. We need to be able to detect all malignant behavior, all malignant cells, and kill or inactivate all the cells that express it. This is a vastly simpler problem.
Can you give me an example to illustrate what you mean that detection is a much simpler problem?
Let's consider one component of malignant behavior, cell proliferation. Cell proliferation is a hugely complex process involving a large number of highly coordinated biochemical processes and interacting networks of genes and proteins. As we discussed previously, a very large number of genetic and epigenetic alterations can cause a cell to proliferate. The complexity is just staggering. Yet all pathways converge and use evolutionarily conserved normal cellular machinery to carry out DNA synthesis and cell replication. As a practical matter, the detection of one or two proliferation-specific proteins can enable the comprehensive and specific detection of cell proliferation. Cell proliferation is easy to detect.
How about invasiveness?
Invasiveness is easy to detect, but hard to inhibit. A small number of proteins and biomarkers should be sufficient to enable the comprehensive detection of invasive processes. The exact number is unknown, but it is probably less than 10. The information required to comprehensively detect invasive processes is far less than that needed to inhibit invasive processes. The modularity and interconnectedness of gene expression networks causes the expression of many of the proteins related to invasiveness to be highly correlated. The prominent role played by non-malignant stromal cells in the tumor cell microenvironment confers robustness and stability to the molecular signatures of invasiveness. A common group of proteins are expressed in physiological invasive processes such as placental implantation, wound healing, angiogenesis, and ovulation. These include urokinase, UPAR, activated MMP2, activated MMP9, MMP14, HSP47, heparinase, plasmin, along with many others. The many factors that are concurrently expressed make invasive processes easy to detect, but difficult to inhibit.
Can you explain in more detail why invasiveness is difficult to inhibit?
A large number of proteases are involved in digestion of extracellular matrix proteins during invasive processes. Inhibiting invasiveness would require the inhibition of all or many of these enzymes, which would be a daunting challenge. This explains the repeated failure of broad-spectrum MMP inhibitors as anti-cancer agents in clinical trials, and the difficulty of completely shutting off angiogenesis. However, redundancy of the mechanisms of invasiveness has its limits. For example, the combination of MMP inhibition and plasminogen deficiency completely prevents wound healing in mice, impairs placental implantation, and causes embryonic lethality.
Is there any single protein or biomarker that is specific or diagnostic of invasiveness occurring?
No. No protein or biomolecule is currently known that is absolutely characteristic of invasiveness. A pattern or combination is needed to detect invasiveness. The same holds true for the detection of malignant behavior. The detection and targeting of malignant behavior requires the detection of a pattern of normal cellular machinery related to proliferation and invasiveness.
Could you explain in more detail what you mean by a pattern?
A pattern is a set of at least two different types of proteins or other biomolecules that are expressed by malignant cells and/or their microenvironment. For the pattern to be present, all elements of the pattern must be present. For example, the pattern comprised of two proteins, A and B, is present if and only if both A and B are present. The information is in the pattern, not the individual elements that comprise the pattern. The detection of pattern AB is logically equivalent or isomorphic to asking two connected questions, or a compound question. Is protein A present, and is protein B also present? In receiving the answers yes to each of the questions.
So why are patterns important?
Patterns provide the only basis for the detection of malignant behavior, and more importantly, for the set of all malignant cells that could evolve. Malignant behavior is a composite property defined by the combination of proliferation and invasiveness and an abnormal context or setting in the body. No single molecular entity can enable the detection of both proliferation and invasiveness, nor provide contextual information. To detect the combination requires the detection of a pattern. Detecting the pattern is logically equivalent or isomorphic to asking the following three-part compound question. Is the cell proliferative? And is the cell engaged in invasiveness? And is the context abnormal? Or, in other words, asking the question, is the cell malignant? As we will discuss later, the third part of the compound question, is the context abnormal, can be dropped, provided that certain conditions are satisfied. Target patterns provide the information needed to detect malignant cells. A set of target patterns, multiple patterns, are required to detect the set of all malignant cells that could evolve. Killing or inactivating the cells that express the target patterns is a separate matter, which we'll talk about later. As a practical matter, each target pattern will be comprised of 2 to 3 different proteins or biomolecules.
Just to be crystal clear, are you saying that the only way to detect malignant behavior is by detecting a pattern?
Yes. Strictly speaking, the only way to detect malignant behavior is by detecting patterns of normal cellular machinery related to proliferation and invasiveness in an abnormal context. This fact has profound practical implications that have been largely ignored by the cancer research community. We need technologies that can enable drugs to detect and target patterns. As a practical matter, the problem can be further simplified since the combination of proliferation and invasiveness is highly restricted and not expressed in most normal tissues of the body most of the time.
Where and when in the body is the combination of proliferation and invasiveness normally expressed?
It is expressed in processes such as placental and fetal development, new blood vessel formation, mammary gland development, the menstrual cycle, ovulation, inflammatory processes such as abscess formation and wound healing. The avoidance of cancer therapy during pregnancy, major wound healing, and infections is standard practice and generally not a significant problem. If we exclude cancer therapy during these normal physiological proliferative invasive processes, then in principle, the set of all malignant cells that could evolve could be specifically detected and targeted on the basis of a set of patterns of normal cellular machinery that effect or reflect the combination of proliferation and invasiveness, or invasiveness and the potential for proliferation, or even invasiveness alone.
Could you explain in more detail why multiple patterns are required and involved?
No single pattern is common to all malignant cells that could evolve. Any genetically encoded protein or biomolecule could be lost or mutated during tumor cell evolution. This means that any given pattern of normal cellular machinery that carries out or reflects malignant behavior could be lost by some malignant cells that could evolve. Furthermore, there are independent, redundant biochemical pathways that can carry out invasiveness. Each must be detected by a pattern. We estimate, based on existing data, that approximately 5 to 10 patterns will be needed to enable the comprehensive detection of malignant behavior and of the set of all malignant cells that could evolve. The exact number remains to be determined experimentally.
There must be a huge number of different patterns of normal cellular machinery related to proliferation and invasiveness. Is it necessary to detect and target all of these patterns?
No. We just need to target a set of patterns that can comprehensively detect proliferation and invasiveness. There are many such comprehensive sets of patterns, but they all convey essentially the same information or message. They are isomorphic.
Would these target patterns be the same for all types of cancer?
Yes. There exists a common and comprehensive set of patterns for all solid cancers. The normal cellular machinery that could carry out malignant behavior is the same for all malignant cells. A comprehensive set of patterns would be able to detect all malignant cells for all types of solid cancers. For example, breast, prostate, lung, ovarian, and pancreatic cancer. This means that one set of drugs could be developed that would be effective against all forms of solid metastatic cancers. The liquid cancers or leukemias could require a different set or additional target patterns. This remains to be determined, but either way, it would be a solvable engineering task to identify and target the patterns.
What experimental evidence is there to support your assertion that there is a common set of patterns or normal cellular machinery that carries out malignant behavior for all types of cancer cells?
The evidence is extensive. Without exception, all malignant cells examined use a core group of evolutionarily conserved proteins to engage in DNA synthesis and cell proliferation, such as MCM proteins and DNA polymerases. A common group of normal proteins involved in invasiveness are also expressed by different types of cancers. For example, MMP2 and MMP14 proteins, or their messenger RNAs, were found to be overexpressed in 32 out of 32 breast cancers, 14 out of 14 lung cancers, 26 out of 26 thyroid cancers, 15 out of 15 glioblastomas, 18 out of 18 prostate cancers metastatic to bone, 102 out of 102 bladder cancers, 10 out of 10 melanomas, 24 out of 24 colon cancers, 18 out of 18 head and neck cancers, 17 out of 18 ovarian cancers, 23 out of 24 oral squamous cell carcinomas, and 23 out of 27 pancreatic cancers. Activated MMP2 was detected at high levels in 33 out of 33 pancreatic cancers, 34 out of 36 lung cancers, 265 out of 269 colon cancers, 31 out of 32 breast cancers, and 8 out of 8 malignant fibrous histiocytomas. Many other normal proteins related to invasiveness are similarly overexpressed in a wide range of cancers. These include urokinase, UPAR, plasmin, emprin, a number of different matrix metalloproteases, fibroblast activation protein, heparinase, HSP47, procollagens, osteospontin, laminin 5 gamma 2, and many others. The experimental evidence is telling us that while every patient's cancer is unique at the genetic and epigenetic level, they all use the same set of normal cellular machinery to engage in malignant behavior. It's important to emphasize that no amount of evidence can prove this point, as a matter of logic. The task ahead is to identify a comprehensive set of target patterns related to the normal cellular machinery of proliferation and invasiveness that are absent from normal tissues. While comprehensiveness cannot be proven, the absence of comprehensiveness can be proven.
What would be involved to identify a comprehensive set of target patterns that could detect all malignant cells that could evolve in the body?
The proteins and biomolecules that carry out or reflect proliferation and invasiveness are largely known. We can list them. What we need is detailed information about the distribution of these proteins in normal tissues and in common non-cancerous conditions such as diabetes, hypertension, coronary artery disease, and rheumatoid arthritis. We need this information so that we can select a set of target patterns related to proliferation and invasiveness that are absent from vital normal tissues. Each pattern will be comprised of 2 to 3 different proteins or biomolecules. Since anti-cancer therapy will not be given during pregnancy, episodes of major wound healing, and during infections, target patterns can be present in these settings. The objective is to select a set of target patterns not expressed by vital normal tissues, such that any malignant cell or its microenvironment will express at least one of the patterns in the set at some point in time.
Normal tissues do not engage in malignant behavior. So why do we need to examine normal tissues to select this set of target patterns?
The reason is that biological systems are degenerate. The same protein can have different functions in different settings. For example, urokinase plays a key role in many invasive processes, as I've mentioned. However, urokinase is also expressed in the normal kidney, which is not engaged in invasiveness. If we want to use a target pattern that contains urokinase, then the second element of the pattern must be absent from the kidneys, so that the pattern is absent from the kidneys. Otherwise, we would get kidney toxicity when we target it. As discussed earlier, a pattern is present if and only if all elements of the pattern are present.
What would it take to identify a set of target patterns that could detect all malignant cells that could evolve in the body for all types of solid cancers?
About 2 years, $10 million, and a focused, goal-oriented project. No new technology is needed. The benefits would be immense. The patterns would provide the information needed to detect malignant behavior and the set of all malignant cells that could evolve. The currently unsolvable problem of curing or controlling cancer would be reduced to the solvable task of detecting a small number of target patterns and destroying cells that express at least one of these target patterns.
What is needed to destroy malignant cells detected by these target patterns?
We need drugs that kill or inactivate cells if and only if the cells or their microenvironment express the pattern. We call this pattern recognition tumor targeting, or PRTT. Targeting specificity must be for the pattern, not for the individual elements of the pattern. The objective of PRTT is not to inhibit the machinery that carries out malignant behavior. It is to kill or inactivate malignant cells. The pattern provides the information needed for detecting malignant cells. The mechanisms of cell killing will generally be unrelated to the particular pattern. Redundant mechanisms of cell killing or inactivation are required. Resistance could develop to any given drug or virtually any particular mechanism of cell killing. However, it is possible to select a set of drugs for which the development of resistance is too improbable to be of clinical significance. In other words, as a practical matter, the drugs would be able to kill any malignant cell that could evolve.
How does pattern recognition tumor targeting differ from using a combination of drugs that target the elements of a pattern?
PRTT is fundamentally different from using combinations of conventionally targeted anti-cancer drugs. PRTT-based drugs are multifunctional and require all elements of the target pattern to be present in order to be toxic to the cells. By contrast, a combination of conventional drugs kills cells that express any elements of the pattern. Consider the pattern AB, comprised of two proteins, A and B. Let's assume A is a marker for cell proliferation and B is a marker for invasiveness. PRTT-based drugs would only kill cells that express both A and B. In other words, only cells that express the combination of proliferation and invasiveness would be killed. Cells that express A alone or B alone would be spared. Normal bone marrow cells proliferate but are not invasive and would be spared. By contrast, a combination of conventionally targeted anti-cancer drugs would target and kill cells that express A alone, B alone, and AB. This would lead to a lack of specificity and bone marrow toxicity.
Can you summarize the key requirements for the specific cure or control of cancer?
In practical terms, the cure or control of metastatic cancer will require multiple drugs administered in combination targeted to abnormal patterns of normal cellular machinery that effect or reflect malignant behavior. A sufficient number of patterns must be targeted such that the probability of a malignant cell evolving without at least one pattern is clinically insignificant. Redundant mechanisms of cell killing must be employed. We estimate that approximately 5 to 10 drugs in combination will be required. These drugs would be the same for all types of solid metastatic cancers. A focused, well-funded, coordinated engineering enterprise is needed to identify and select a comprehensive set of target patterns, to develop PRTT platform technology to enable drugs to kill cells if and only if the cells or their microenvironment express the target pattern, to develop PRTT-based drugs targeted to specific patterns, and finally, to develop a set of PRTT-based drugs that can be given in combination for the specific cure or control of all types of solid metastatic cancers without significant toxicity to the patient.
Earlier you mentioned that there are some special cases like brain cancer that could evolve but be cured by locally abolishing the potential for cell proliferation without the need to target patterns. Could you elaborate on this concept a little further?
Sure. Cell proliferation is an absolute requirement for the progression of cancer, but is virtually absent within most regions of the normal developed brain. Abolishing the potential for cell proliferation within tumor-bearing regions of the brain would cure brain cancer. A single brief exposure to certain drugs can permanently abolish the potential for cell proliferation without killing cells or significantly damaging non-proliferating cells. The drugs work by inactivating normal cellular machinery that is required for cell proliferation. The local administration of such drugs into the tumor-bearing regions of the brain could provide a therapy for the treatment and cure of brain cancers and metastatic lesions to the brain and central nervous system. Delivery of the drug to all cancer-bearing regions of the brain is essential. Multi-site drug delivery would be needed into the brain, but this is a solvable engineering task well within the scope of existing technology. Since safe techniques exist for the direct infusion of anti-cancer drugs into the brain, and the drugs exist, clinical trials could begin within 2 to 3 years of funding. This approach could provide an effective means to treat and cure brain cancer and metastatic lesions to the brain, regardless of the underlying genetic and epigenetic alterations.
Can you explain in more detail what you mean by abolishing the potential for cell proliferation?
A drug that abolishes the potential for cell proliferation will permanently prevent cells from being able to proliferate without killing the cells, regardless as to whether or not the cells are actively proliferating at the time of drug exposure. This is important because most malignant cells are not actively proliferating most of the time. This is very different from using a drug that kills proliferating cells or that actively inhibits cells that are engaged in proliferation.
Isn't cell proliferation needed to maintain blood vessel formation in the brain?
Yes. Cells and blood vessels proliferate about once every 1,500 days. However, there's good evidence that circulating bone marrow-derived endothelial stem cells can regenerate and repair blood vessels. So the inhibition of cell proliferation in blood vessels is not expected to be a problem. However, drug administration would need to be delayed to allow time for healing after brain surgery.
What do you see as the biggest challenge to be overcome to develop this approach for the treatment and cure of brain cancer?
By far, the biggest challenge is funding. $10 million in about 2 to 3 years would be needed before clinical trials could begin. Several years ago, a number of us organized a non-profit project involving 5 major brain cancer centers to develop this approach. Senator Kennedy had requested congressional funding for this brain cancer project. However, his tragic death from brain cancer and changes in the political environment ended the ability to build support for this congressionally directed funding approach.
Do you have any concluding thoughts
Yes. First, I'd like to thank you for the opportunity to appear on the new Cancer Mentality website. Second, I would like to state that there is every reason to believe that cancer can be cured, and that some of the most common types of cancer can be prevented. However, this will take some major changes in the way that things are done. At the scientific level, approaches to the prevention, treatment, and cure of cancer need to be logically consistent with the reality of nature, that cancer is an enormously diverse, unpredictable, stochastic evolutionary process. This implies, contrary to conventional wisdom, that the essential features of treatment for the cure or control of cancer must be independent of the underlying genetic and epigenetic alterations. To consistently cure or control cancer, it is necessary to kill or control the set of all malignant cells that could evolve. This will require targeting a comprehensive set of patterns of normal cellular machinery related to proliferation and invasiveness. The same set of patterns will apply to all types of metastatic solid cancers. The identification of a comprehensive set of target patterns and the development of technology to kill cells that express these patterns is a solvable engineering task. It will take a well-funded, well-coordinated engineering enterprise. The technology exists. What we need is the courage, will, and determination to listen to what all the scientific evidence is telling us and change course, or, at a minimum, to pursue a parallel course and develop cancer therapy that is independent of the underlying genetic and epigenetic chaos of cancer. I believe that the requirements for the cure of cancer that we have discussed can be met. It is time to establish a major multi-institutional project to develop a set of drugs for the specific cure of cancer. Cancer patients and their families deserve no less. Thank you.